Assalamualaikum. My name is Raina Nadla. I'm from DBT4A. I will do a video about bacterial identification visual lab. Uh, the purpose of the lab is to familiarize you with the science and technique used to identify the different type of bacteria based on their DNA sequence. Not long ago, DNA sequencing was a time-consuming, tedious process with readily available commercial equipment and kits. It is now a routine. The techniques used in this lab are applicable in a wide variety of settings, including scientific research and forensic lab. There are four basic steps. The first one is prepare a sample from a patient and isolate whole bacterial DNA. Second, make many copies of the desired piece of DNA. Third is sequence of the DNA. And the last one is analyze the sequence and identify the bacteria. The first step is sample preparations. Click one of the bacterial colony in the dish and transfer the bacterial colony into the microcentrifuge tube. Add a digestive buffer to the microcentrifuge tube. Wait for several hours. Move the microcentrifuge tube to the water bath to heat it. Loading the microcentrifuge tube to the centrifuge. Transfer the supernatant to the PCR tube. The sample preparation is complete and we go to second part, which is PCR amplification. Add the master mix solution to the PCR tube. And add control DNA to the positive control PCR reaction tube. Okay, next, add water to the negative control PCR reaction tube. Loading the PCR tube. The PCR tube has successfully been loaded in the PCR machine. We go to the next process, which is PCR purification. Add a buffer solutions to the column. Add a PCR product to the column. Put the tubes on the ice. And loading the column into the centrifuge. Adding the counterbalance. And wait for 15 minutes. Adding a new collection tube. And inverting the column. Add a buffer solution to the inverted column. Discard the first collection tube. Now I will centrifuge the assembler to collect the DNA into the collection tube. Adding the counterbalance to the and click the centrifuge and wait for 2 minutes. Discard the column. Okay, we go to part 4, which is DNA sequencing. Prepare for sequencing. Add a distilled water to the purified PCR product. Add a purified PCR product to the strip tubes. Loading the strip tubes. Each green and blue tubes contain a sequencing board consisting of buffer primers, DNA polymerase nucleotide of fluorescence stacked emitter in suitable proportions. The PCR product from the previous step is added to each tube and another PCR is run. This time the aim is not to produce identical copies of DNA but many copies of variable length. The animation illustrates what is happening in one tube containing the primer SIG51R. Each DNA strand binds the primer at one end and will have a fluorescence stacked emitter at the other end. Okay, next we go to the DNA sequencing. Loading the tray. Click the inner gray bar above the the orange thread to view the DNA sequencing animation. This is done by using an automatic sequencer that performs gel electrophoresis on the DNA in each tube. Electrophoresis is a method to separate molecules based on differences in size. The sequencer used in this lab has a thin capillary tube attached at one end to a string mechanism that contains a buffer solutions. The tube is filled with the buffer solutions and the other end inserted into one of the tubes containing the DNA pieces. Then an electric current is applied so that the end of the tube in contact with DNA has a negative charge and the string end a positive charge. We go to the last part, which is sequencing analysis. On the NCBI website's list page, under the Enter Query Sequence sections, paste your sequence data from step 1 into the box labeled Enter Accessions Number. Under the Choose Set Database section, Standard Database, Nucleotide Collections, When you are ready, click on the blast button further down the page. Okay, that's all.